I'd like to welcome everyone today to this seminar series. My name is Valerie Odero-Maro. I'm the Executive Director for the Center for Health Disparities Research and Innovation here at Morgan State University. I would like to welcome you to today's event. It's the second in a series of educational programs that are actually a result of collaborations between multiple groups at Morgan State University and Johns Hopkins University. So these programs are designed to increase community awareness on topics related to Alzheimer's disease and related dementias. It's also to provide information about treatments for individuals with memory loss and to help highlight the importance of diverse participation in research. This event is presented by the Morgan State RCMI program, Ascend, and the School of Computer, Mathematical, and Natural Sciences. It's co-sponsored by Morgan Cares, the Johns Hopkins Alzheimer's Disease Research Center, and the Johns Hopkins Alzheimer's Disease Resource Center for Minority Aging Research. And so this seminar is open to the community. And our speaker today will be Dr. Barry Greenberg. Corinne, do you want to introduce Dr. Greenberg?
Sure thing, no problem.
Thank you.
Hi, everyone. Welcome. We're so happy to see so many of you here today. Barry, if you could go to the next slide, please. So today's speaker is Dr. Barry Greenberg, who is an associate professor of neurology at Johns Hopkins University School of Medicine. He's been involved in Alzheimer's disease research and drug discovery since 1985, holding a series of positions internationally in the United States, Sweden, and Canada, including with biotechnology and pharma industry, as well as in academia. He was chair of the National Institute on Aging's International Committee that was charged with strategizing the future structure of U.S. Alzheimer's Disease Research Center networks in order to augment their capacity to develop effective treatments and preventions for Alzheimer's disease and related disorders. He's recently become editor-in-chief of the Alzheimer's and Dementia Journal, and he brings a really multifaceted perspective to Alzheimer's disease, with an international reputation drawn from hands-on experience in basic research through human studies. So today, Dr. Greenberg is going to be speaking on aducanumab, and I will go ahead and turn the floor over to him.
So some information, if you have questions, put them in the chat and we'll answer them at the end, because we intend to have a healthy discussion at the end of the talk. So please put your questions in the chat. And if you're not speaking, please make sure to mute your microphone. Thank you.
I'd like to thank you all for that introduction, and also thank the sponsors, Morgan State, Morgan Cares, the Johns Hopkins Alzheimer's Disease Research Center, and the AD Resource Center for Minority Aging Research. As someone who's been involved in the Alzheimer field, Corinne tipped it, since 1985, and a large part of that has been on drug discovery related to amyloid. This topic on aducanumab is one that is quite close to my heart, and I appreciate the opportunity to be able to speak on this. I'll give you a bit of an overview. This talk over the next 35 minutes or so is going to be split into three parts. There's background, there's the controversies, obviously, because controversies is part of its title, and then future directions and final thoughts. And for background, I'll be describing current approaches to developing therapies for Alzheimer's disease, the drugs that have been approved for Alzheimer's disease, the aducanumab clinical trials, the latter trials that went into the FDA considerations, and then the FDA approval process. Under the controversies heading, we'll talk about the FDA decision on aducanumab, financial considerations, costs and coverage, and issues about making the personal decision of whether to go on or not, go on the drug or not. So in the background, here we go. Broadly speaking, there are two classes of medications for Alzheimer's disease: symptomatic therapies and disease-modifying therapies. Symptomatic therapies improve symptoms, as you might anticipate from the title. They do not treat the primary pathology. And these are measured through measures of cognition, brain function, and activities of daily living. Disease-modifying therapies are a bit of a different flavor. They are meant to prevent or slow the progression of the disease itself in earlier pre-symptomatic stages by targeting the pathology or processes that cause Alzheimer's disease. These may take longer than symptomatic drugs to show effect. They may be difficult to see in an individual patient, and I'll be speaking more about that a bit later. The clinical trials take longer and are more difficult than for symptomatic drugs, and to date, the majority of trials have targeted amyloid and more recently tau. So why amyloid and tau? On this slide, you see a picture of Dr. Alois Alzheimer. And he spoke, I can't see my title, it was in the Tübingen, I can't see the title. It was a Tübingen meeting of the Southwest German psychiatrists in 1906. This was in November of 1906, 116 years ago almost. And he presented a case report study of his patient, August Dieter. It was a 56-year-old woman who had an aggressive, rapidly progressive form of dementia with agitation. She was a difficult patient. And when she died under his care, he did an autopsy on her brain to have a look at what was going on. And these were the sorts of structures that he drew, that he found in her brain that hadn't been seen before. And if you look under more modern techniques, these are silver-stained structures of a tissue slice from an Alzheimer's diseased brain. These are the neurofibrillary tangles that are comprised of the neurofibrillary protein tau, neurotubule binding protein tau. And these are what you see Dr. Alzheimer quite accurately drawing in his presentation and his case report that went into his first publication. And in addition to these structures, you can also see these are intracellular structures within the affected nerve cells, neurons. Extracellularly, there are amyloid deposits, also referred to as amyloid plaques, and these are comprised primarily, but not exclusively, of the amyloid protein that has been called A-beta. An important point to consider is that Alzheimer's disease has a long pre-symptomatic phase during which these pathological structures begin to form. Now, this is an opportunity for treatment and prevention. Nearly all of us have amyloid entangles, some amyloid entangles in our brains once we reach middle age. It's the amounts and the rates of accumulation which contribute to whether we develop Alzheimer's disease. Not only the amounts and the rates, but also the other processes that respond to the formation of these pathological structures. Inflammatory processes have come into attention recently. But these are thought to be the precipitating events in the pathological cascade that leads to the cognitive symptoms of Alzheimer's disease. Now, for symptomatic drugs, the approval is based on demonstrating higher cognitive test scores relative to placebo, or a sugar pill in the case of these drugs, because they're all pills, in patients who are symptomatic with mild to moderate dementia. And these names, these are the 4 drugs that have been approved. They may be familiar to some of you: donepezil, galantamine, rivastigmine, and Namenda. Excuse me. The treatments are aimed at the brain chemistry that's associated with Alzheimer's disease. They don't change the rate of decline. Patients who tolerate these drugs, and they're tolerated, there's some side effects that have to be watched out for, but is there a question? Patients who tolerate the drugs are improved for a limited period of time, but then they decline at the same rate as patients who are either not on the drug or as they would have been declining if they had never taken the drug. The disease-modifying drugs are a bit different. Here, participants are recruited into trials who are mildly impaired. It's thought with pretty good evidence that treating individuals with moderate to advanced disease may be too late. Rather, treating pre-symptomatic patients would be even better. Identifying these individuals and exposing them to the possible side effects of drugs before symptoms occur is a problem. So it's an extremely active area of research to be able to identify these individuals reliably and, of course, to develop drugs that are going to be safe enough that they can be taken that early and for the long period of time that would be required. It's important to determine if the participants have the brain abnormality that the drug being tested is intended to treat. Most recent studies have been enrolling individuals with a specific amount of amyloid or tau pathology in their brains. It was probably the most embarrassing moment in the history of Alzheimer's disease drug development when the first trials that were using drugs targeting brain amyloid were concluded and then analyzed. And it was during the course of those trials that the ability to image amyloid in the brain of a living subject was really being developed. And it was determined post hoc at the end of the trial that about a third of the patients who were in that trial who were enrolled because of cognitive dysfunction, cognitive deficits, memory problems, did not have any amyloid in their brains. It is possible to have these symptoms without having Alzheimer's pathology. So we're not going to make that mistake again. The subjects that go into trials are most definitely diagnosed with having the brain pathology that is being intended to treat. And then it needs to be determined if the medication is slowing the progression of the disease. And for this, measures that reflect change in cognition and daily functions are used in combination with measures that reflect the change in the brain pathology, so-called biomarkers. And that can be imaging, brain imaging biomarkers, or fluid biomarkers taken from the cerebrospinal fluid that's obtained by lumbar puncture or spinal tap. And more recently, developments are in full swing to be able to observe these biomarkers in blood, in plasma. Until last June, Namenda was the last drug that was approved for Alzheimer's disease, and that was in 2003. We're talking 18 and a half years ago. And that's despite hundreds of clinical trials that have involved hundreds of thousands of patients and billions of dollars spent since then. This is a hard disease to treat. No disease-modifying drug was approved until 2021, last June, and that's aducanumab, which has been branded by the name Aduhelm. This is a monoclonal antibody treatment to remove brain amyloid. Now, the question is the billions-dollar question. Is Aduhelm the first truly disease-modifying drug? And I'll give you a bit of a spoiler alert for the rest of my talk. Doesn't mean you should turn it off yet. We don't really know yet. So these were the aducanumab so-called phase 3 clinical trials. I'm not going to describe what phase 1, phase 2, and phase 3 are, but phase 3 trials are the so-called pivotal trials, the large trials that are used to accumulate the data that if the trial is successful or arguably successful, the data can be brought to the Food and Drug Administration, the FDA, for consideration of approving the drug. And there are two identical 78-week studies, year-and-a-half-long studies, that were conducted simultaneously. As is the tradition in the drug world, these trials are named. These two trials were Emerge and Engage, and both of them had over 1,600 subjects. It was double-blind placebo-controlled trial, so half the subjects were on drug, half the subjects were on placebo. This was conducted at 348 study sites in 20 countries. This is a big deal. The primary participant enrollment criteria were mild cognitive impairment or mild dementia, and a significant amount of amyloid in the brain is measured with PET scans. The treatment strategy was a monthly, hour-long intravenous infusion, either of drug, aducanumab, or placebo, in this case, saline. The dose varied over time, and some of these variations were pre-specified. So individuals were put on escalating doses from low dose up to high dose, with the high dose depending upon certain attributes of the subjects, which I won't go into at this point. Other variations were by protocol revisions that were done on the fly during the course of the study based on what was learned in the study and based on what was learned by analysis of previous studies while these were being conducted. And these protocol revisions led to some statistical difficulties that actually plagued the analysis of these trials when they were being studied by the FDA. The measures of effect there were two, primary and secondary. The primary is the important one. That's the one that is what the subjects who have the disease experience. And this is what you need to improve for an Alzheimer's disease drug, whether it be symptomatic or disease modifying. These are daily functions that are impacted by cognition, and the measure that was used in this case was the so-called clinical dementia rating scale, also abbreviated CDR, and the form that was used was the CDR dash sum of the boxes. The secondary measure was to demonstrate that the drug was reaching its target. And this was a measure of amyloid burden that was assessed by PET scans. This is not part of the experience of the disease, but it's part of the pathology. As has been said numerous times, you have no idea if you have amyloid in your brain unless you had a brain scan. No one ever walks into his neurologist and says, "Doctor, I've got a boatload of amyloid in my brain." But they do walk into their neurologist and say, "Doc, I've got some memory problems. I've got some cognitive problems." So it's that primary effect which is important. The secondary effect demonstrates disease modification. Now, these are the primary findings from the two trials, Emerge and Engage, and this is an important slide to spend a little bit of time on. These analyses were based on subjects who completed 18 months on the highest dose, and that was approximately half of those who were enrolled in the studies for reasons that I can elaborate on later. But suffice to say, about half of the subjects completed the trial, and it was their data that went into these graphs and these considerations. All right? What you see is the drug worked in terms of reducing brain amyloid. It was significantly reduced over time in both studies. So this is amyloid PET. This is the Emerge study. This is the Engage study. The solid line were the subjects who were on placebo. The levels of amyloid did not change in their brains over the year and a half on the study. The dotted lines are the subjects who were on drug. And you can see precipitous drops in the levels of brain amyloid during the course of the study. Drug was getting into the brain. It was removing amyloid as it was designed to do. There's a however coming up. There was a small but statistically significant benefit on the cognitive measures, the CDR, in only one of the studies, Emerge, but not in Engage. Here, to orient you, the higher the score on the CDR, the worse things are. So a lower score is better, a higher score is worse, and you can see that the patients on placebo, both in Emerge and Engage, were getting worse during the course of the study. The patients on drug did better than those on placebo. It's this difference which was statistically significant in Emerge. This difference in Engage was not statistically significant. But there's a couple things that you really need to look at on this part of the slide. First of all, the patients who were on drug still got worse during the year and a half. Their CDRs were going up during the course of the study. So this drug reduces progression but doesn't stop it. It slows progression. The other thing to appreciate here is look at the two studies, look at the treatment effects. They're essentially identical. Right? We get to the same CDR score across these two studies. What really differs is the decline in the placebo group, which was greater in Emerge than it was in Engage. And this is what led to some serious considerations of whether or not there was a positive study or not. Which of these studies was believable? They were identical for treatment. They only differed in terms of the placebo behavior. All right, we'll move on. Now let's talk about the FDA approval process. An independent advisory committee meets with the Food and Drug Administration and the drug sponsor, reviews the study findings, votes, and makes its recommendations. This, in the case of these sorts of drugs, is the Peripheral and Central Nervous System Drugs Advisory Committee. Also referred to in the press, you may have seen the term ADCOM. It's the ADCOM who met with the FDA. And the ADCOM consists of experts in the field, including neurologists, psychiatrists, neuroscientists, statisticians, whoever is needed to analyze the study data. And the FDA reviews the ADCOM recommendations and then makes a final judgment based on the weight of the evidence. That's what happens at the FDA. Now we're going to talk about the controversies. That's all the background. The AdCom met on November 6, 2020. That's 116 years and 3 days after Alzheimer made his first presentation in Tübingen of his first patient. And the AdCom voted almost unanimously against approving this drug based on the balance of risks and benefits. Now, I haven't told you about the risks. Brain swelling and other abnormalities in brain blood vessels were found in approximately 40% of the treated subjects. Most of these were minor, but not all. And
Dr. Greenberg, before you go on, there's your audio is going in and out.
oh, I'm
So probably
sorry. I don't know what to do about that other than get closer. Is that
so,
better?
yeah.
Okay.
Yeah, so we'll go. Thank you.
My apologies. Most of these abnormalities were minor, but not all, and this requires regular brain imaging to monitor for these conditions and adjustments in dosing when they occur. The benefits, the AdCom and the FDA's own statistician agreed that the evidence was unclear that the drug improved the primary outcome, the CDR, based on the results of the conflicting studies. However, on June 7th, we're talking 7 months and a day later, rather than requiring a third confirmatory trial as was expected, the FDA granted a so-called accelerated approval based on the evidence that the drug reduced amyloid in the patients who completed the trials, with the justification that amyloid lowering as a so-called surrogate marker was reasonably likely to harbinger future clinical benefit. Now, this is despite the fact that the FDA had made the statement that they were not going to base approval on a surrogate marker. And that was said to the AdCom before they met in November, and this decision was never discussed with the AdCom, so you can see how this became pretty controversial. An additional trial was required as part of this approval, although the drug can be marketed while that study proceeds. So, June 7th, 2021, landmark day. Aduhelm is approved in the United States as the first disease-modifying drug for prescription use. And this was an OMG moment for the field. I'll have more to say about that. Nice package. We have some issues. A link between amyloid reduction and a meaningful functional outcome in symptomatic patients has never been convincingly demonstrated. Most experts in the Alzheimer field have stated that the AdCom's recommendation was correct in disagreement with the FDA approval. Several major medical centers and the Veterans Administration, the VA, have decided not to make Aduhelm available in their institutions. Among those major medical centers is Johns Hopkins. To date, other international drug regulatory agencies have refused approval or marketing authorization. This drug is approved only in the United States. And its use will require substantial integrated medical infrastructure, including, but not necessarily limited to, expert multidisciplinary clinicians skilled in early Alzheimer diagnosis, which is not simple, and comorbidities, infusion centers with access to brain imaging and its interpretations, which requires radiologists, PET imaging or lumbar puncture, spinal tap capabilities, with associated expertise to interpret the biomarker results, genetic counselors, sociologists, and so on. So this is going to be a substantial requirement to bring this into medical practice. And further problematic were the demographics of engaged and emerged trial subjects. And this was a slide that was taken from a presentation by Jennifer Manley at Columbia University last September, but this has since been published, so I don't have to be worried about stealing it anymore. To orient you to this table, these are the demographic parameters of the subjects who were in the trial, whose data went into the consideration by the FDA. These are the numbers and percentages who were on placebo, aducanumab low dose, aducanumab high dose. As I said, these data represented about half the subjects who enrolled in the study, so the total number of subjects was 1,638. Of those, 1,285 were white. 11 of them, less than 1%, were Black or African American. 128 of them, less than 8%, were Asian. 67 of them, 4%, were Hispanic or Latino. Now, we know that the prevalence of Alzheimer's disease, its genetics, its risks, its co-pathologies, and possibly biomarkers differ among these races. So what have we, now this was not by intent, so I'm not trying to cast aspersions at Biogen. This is just data, this is fact, all right? So what have we learned about Alzheimer's disease? We've learned the effect of this drug on people who look like me. And I'm not talking about an old ugly guy who you see on the screen. I'm talking about a non-Hispanic white of European descent. That's what we've learned from this study. Now, in terms of public health, what is the risk benefit? Who should be exposed to the drug? The answer to that is we don't know. Meanwhile, 3 follow-up studies are planned by Biogen. This is all good. The EMBARK study, the iCare ADUS study, and the ENVISION study. EMBARK is a phase 3B redosing study that was announced last July. This is open label. That means everybody who's in the study is going to be taking drug and they know it. So it's not placebo-controlled. It's a high-dose treatment of all eligible participants who had previously been enrolled in aducanumab clinical trials, whether they were on placebo or drug. And they're planning to enroll approximately 2,400 subjects for 24 months treatment, 28-week follow-up, and assess for long-term safety and efficacy, including biomarker outcomes. It's all good, but keep in mind the demographics will be the same as you saw in that last table. There's iCare ADUS. This is a phase 4 observational real-world study that was also announced last July. Also open label. These are individuals who will go on drug and know it. And the criteria will be based on the FDA-approved prescription label, the prescription requirements. Real life, clinical. Designed to evaluate the safety and long-term effectiveness in clinical practice. They're aiming for 6,000 subjects enrolled over 4 years from 200 U.S. sites and monitoring for up to 5 years. And here they're targeting 16% of the subjects to be enrolled from Black or African American or Latinx communities, populations. Now, that's about 3 times the percentage of Black African Americans and Latinx individuals in the clinical trials. I'm not sure where the Asians went. And then the ENVISION study. This is the post-marketing phase 4 confirmatory study that was mandated by the FDA. This was announced last January. This is a placebo-controlled double-blind trial. So subjects will be either on drug or on placebo. They're targeting 1,500 individuals. It will have a similar study design, outcome measures, and duration as the phase 3 trials. Similar enrollment criteria, except they're aiming for 18% Black, African American, and Latinx participants. Screening is going to begin next month. The completion date is anticipated 4 years later. Now let's talk about costs. The drug has been priced at $56,000 per year. And it will cost an additional approximately $30,000 per year for related medical procedures. There's a big outcry over this. We're talking about lots of people needing drug, and that's an expensive drug. Last January, with some fanfare, Biogen decided to reduce the price, or announced that they were reducing the price by half to $28,000 per year. Lost in that announcement, but if you read a little bit deeper into it, that was for subjects weighing 74 kilograms or less. That's 163 pounds. So if you consult the good Dr. Google, you'll find that the average weight in the United States for an individual 60 years old or older is 195 pounds for men and 166 pounds for women. So this will be applicable to about half the women and less than half the men who would be taking that reduced rate. So that's really not a 50% reduction when you think about it. Several U.S. private health insurers have announced they will not cover Aduhelm due to clinical efficacy being unestablished. A large majority of qualifying patients will be on Medicare for obvious reasons. The decision on Medicare coverage is due this month. I've actually been watching the news all day today to make sure it didn't come out this morning because I would have had to have changed some slides at the end here. It hasn't. It's due later this month. But if it's covered by Medicare, there will be health equity issues related to access and affordability. Why? Infusions are covered by Medicare Part B, and that's a 20% copay, which means it's going to cost out of pocket either $5,600 or $11,200 per year, depending on how much you weigh. And that's equivalent, and just to bring the example home, to either 12% or 24% of the median household wealth among older U.S. black adults in the country per year for every year they're taking the drug. Yeah. CMS, the CMS decision also adds to some of the controversy. CMS is the Centers for Medicare and Medicaid Services. This is Medicare. Their draft recommendations were released last January, and they limit aducanumab coverage only to subjects who are participating in randomized controlled trials. That limitation would cover Biogen's Envision study, the one that was mandated by the FDA, but not their Embarker eye care studies. This was roundly criticized by Biogen and some advocacy organizations, but praised elsewhere as accelerating knowledge and health equities required for delivery of successful therapies into the public health realm. And I would refer you to this article. Anybody who wants to see this article, just let me know and I'll be happy to send it along. The Institute for Clinical and Economic Review, ICER, doing what they do, assigned a rating of insufficient whether aducanumab provides a net health benefit. And their cost analysis paradigm suggested that the small health gains justified pricing at $3,000 to $8,400 per year, not $56,000 or $28,000 per year. And in this article that was recently released in a publication called Health Affairs Forefront, I lifted a quote. There is need for more FDA-CMS coordination prior to product approval, especially for drugs expected to have large budgetary and clinical consequences. Let's talk about the burdens. The burdens of going on drug or the burdens of not going on drug. You can anticipate that that's the next slide. It has been estimated that patients and care partners, and every patient needs a care partner for this one, should expect to commit at least 16 days a year to complete their evaluations, investigations, and their infusions. And that's if nothing goes wrong. Substantially more than 16 days in the event of any emerging adverse events, like we're seeing in the 40% of the treated patients in the clinical trials. Unresolved is when to stop the drug. Remember, it's not a cure. It only slows decline for some. And slowing decline rather than stopping decline, that's an unclear clinical effect in an individual patient. How does the individual patient know that he's declining more slowly than if he weren't on the drug? Unless there are subjective reasons why that individual may feel that he or she is benefiting from the drug, and there is anecdotal evidence that that has occurred in some of the people who are in the clinical trials. But it's unclear. So the questions, three rhetorical questions. How long is treatment acceptable to determine whether it's working? When to stop it if it is working? Remember, these individuals are going to progress past the phase of the disease where the drug is effective. And when to stop it if it's not working. What about the burdens of not going on drug? These are also very difficult. It leaves the perception that you're not doing everything you can. If you or someone you care for has been diagnosed with Alzheimer's disease, you or that person are not taking the drug. It's a drug that is being promoted for its potential benefits. It's being strongly campaigned for by advocacy organizations. And if you haven't seen this on your television screen yet, you're going to see something like it soon. This is from one of the advocacy organizations. This is a powerful ad. Look at that face, right? It says, Alzheimer's patients can't wait. Medicare, don't deny coverage for FDA-approved treatments. So making the decision. The accelerated approval by the FDA was based on a reduction in a biomarker, but there's no requirement to measure that biomarker to manage the therapy. So how do you know that it's actually hitting the target in you or in the person you care for? There's no requirement to monitor that. No presence of pre-existing brain amyloid or genetic testing were required. Now, why do I bring up genetic testing at this point in the talk? I didn't go into it earlier. But according to the FDA statistician's analysis, only subjects carrying the form of the apolipoprotein E gene that's associated with elevated risk for Alzheimer's disease demonstrated positive clinical outcomes. That's about half the people who have Alzheimer's disease carry that form of the gene. So the other half, what if you have the other half? There's no evidence that this drug is going to work for you. So there's no requirement to determine what form of the ApoE gene you have as part of the label. What should you expect if your race or ethnicity differs from the trial subjects? It's a good question. We'll learn from things like the eye care study. We have no potentially important exclusions. Now, I mentioned that there were vascular risks, blood vessel risks. So individuals who were enrolled in the clinical trial were excluded from the trial if they were on anticoagulant therapies. That's not required as part of the label, as part of the treatment that's been approved. In fact, published post-approval analyses have estimated that 92% of patients who currently qualify for the drug would have been excluded from participating in the clinical trials. And the decisions to start and stop the treatment are hard. They should be up to the patients in consultation with their physicians and their care partners, whose lives will also be changed by this, logistically and economically. But the patients need to be fully informed and understand what they're committing to undertake. And I'll refer you to this excellent position statement from the American Association for Neurology that came out last year, and there's an even newer one that came out this year. And again, if you want these articles, let me know. So now let's talk about future directions and final thoughts. Aducanumab is going to be used by some patients, and additional data will become available as a result of that from patients who are currently seeking treatment, from following patients who are enrolled in the original and follow-up clinical studies. So for example, last month at the Alzheimer's disease and Parkinson's disease conference, international conference in Barcelona, March 16th, Biogen had a report where they defined that they showed a continued reduction in the biomarkers of pathology, amyloid and tau, between 78 and 128 weeks, 50-week extension in subjects from the phase 3 trials who were on extended treatment. And that's great. What they didn't mention anything about was whether there were any additional data on cognition or function in these patients. We can anticipate policy development with far-reaching effects. What are going to be the impacts on considerations of drug pricing controls? Medicare has historically paid for FDA-approved drugs. Question is being raised, should any FDA-approved treatment be covered by Medicare regardless of the benefits versus the risks? And who pays for that? Last November, CMS announced that the monthly Medicare Part B premium would rise from $148.50 in 2021 to $170 in 2022. That's a 14.5% increase of people who are paying Medicare Part B premiums. That's a $21.60 per month increase. So you know who's paying for this. So where are we now in treating and preventing Alzheimer's disease? I want to really emphasize this first bullet point. This is a remarkable watershed moment in the history of Alzheimer's disease research. A potential disease-modifying therapeutic agent. The science is solid. The data are solid. We know what we know from these data. Right? Remarkable moment. The controversies are the decisions that are being made at this time in light of those data, which many feel is not complete enough for those decisions to have been made yet. But the science is good. Some patients undoubtedly benefited from aducanumab. We need to understand who, why, and how well, and be able to ask the question and then ask it, if this worked in this trial, who's it going to work for in real life? We need to keep in mind that there are 3 more similar therapies lined up for accelerated approvals. We're going to learn from the similarities and differences between these clinical trials when they're fully analyzed. This is a point from which to build. It's not the goal line. And my parting message. The Alzheimer research field remains committed to learn what it can from this moment and build on that knowledge to deliver safe, effective, and affordable therapies. There's a continued urgency to understand the early phases of Alzheimer's disease and how to treat it. Many biological targets and mechanisms remain to be explored. Different drugs or drug combinations, together with lifestyle modifications, may be appropriate for different individuals and may include aducanumab. We just don't know yet. And this is why continuing and expanding research studies is so critical. It's the ongoing willingness from generous and selfless research subjects to engage in clinical investigations that's the key that will unlock the door to the ultimate achievement of preventing and treating Alzheimer's disease. And with that, I'd like to thank you for your attention, and I will stop my talk and we can open up for discussion.
Thank you, Dr. Green.